students, and welcome to our topic on not darkness, but on what is the topic for today? Our topic this week is genetic engineering. That's right. We've already watched some videos on genetic engineering. It's a fascinating, growing field in not only this country, but around the world today. And if this topic interests you, you might want to pursue a career in genetic engineering. So if it at all sounds interesting to you, let's find out a little bit more about our topic, genetic engineering. So what is it? In the most general terms, genetic engineering is the process in which genes, and we know what genes are, from one organism are transferred into another organism. Now, we could even expand that definition today in terms of not just being transferred into another organism, but even within the same organism. The technology of genetic engineering is growing so quickly and advancing so quickly, it's impossible to keep up with all of the changes taking place even day by day. But for our purposes in this topic, we will consider that genetic engineering is the process in which genes from one organism are transferred into another. And I understand that, again, every day this field of study is changing so quickly that we could add things to the following list. But for our purposes on this topic, we're going to simply limit ourselves to 5 techniques that can be utilized in pursuing genetic engineering today. And so, what are these 5 techniques, broadly speaking? Well, they begin with number 1. It's called recombinant DNA. And we'll talk more about each of these as we go through the lesson today and tomorrow. So, number 1 is recombinant DNA. Number 2 is called gene isolation. Gene isolation. Number 3 is gene targeting. Gene targeting. Number 4 is called transformation. Transformation. And finally, number 5, is regeneration. Regeneration. So these are the 5 techniques we're going to be exploring today and tomorrow. So let's begin with number 1. Number 1 is recombinant DNA. And when we talk about recombinant DNA, we're talking about having a section of DNA from one organism is joined or combined to another organism's DNA. Recombinant kind of sounds like recombination, combining or joining DNA or a section of DNA from one organism into another. Now, if you look at this example diagram here we have 2 different organisms' DNA, and the illustration is showing that we're taking one section from one organism's DNA and joining it into another organism's DNA. And I'm going to show you a video of this. When we're finished with our notes today, I'm going to show you videos of each of these different types or techniques of genetic engineering, because the videos will do a much better job at explaining this and illustrating it than I certainly can do myself. But here's an example of recombinant DNA, a very famous example. It has become a rather famous example of making glow-in-the-dark bunny rabbits. Yeah, how do they do it? Well, Take the glow in the dark gene from a jellyfish and insert it into the DNA of a bunny rabbit. And they've used other animals too, for that matter. And we have produced bunny rabbits that actually glow in the dark. Now, another example is putting a human protein into yeast or into bacteria and allowing the yeast or the bacteria to do the dirty work of reproducing, which then reproduces that human protein DNA in it. And I think you'll find the video on this rather interesting as well. But it's essentially our utilization of a microorganism to be a DNA molecular factory reproducing certain segments of DNA that we can utilize then typically in the healthcare industry in medicines. It's a fantastic technology that we're making use of. And as I have said, and I'll continue to say, when we watch the video segments on this, you'll see exactly what I mean. So I mentioned earlier that the glow in the dark jellyfish has been used in rabbits. Well, it was used in mice too. And so here are glow in the dark mice that have the DNA segments from jellyfish spliced into them. Here's just a funny little cartoon. It says, at harvest time, Ted's ethical objections to the use of frog genes in potato breeding were conveniently forgotten. And what this little cartoon is telling us is this. When a new technology comes out, it's very easy to say, oh, we're opposed to this technology because bad things can happen. And I'm not discounting the possibility that that is true. But when the average person discovers that there is a true convenience to the new technology, then gosh, we kind of forget about the ethical objections that we had in the first place. I mean, this is really silly, this cartoon, but it makes this point. It may be that this potato farmer was opposed, and again, we're speaking hypothetically here. This isn't actually being done, or at least I don't think it's actually being done in the world right now. But the point it's trying to make is that we could have a potato farmer ethically opposed to the use of potato genes being spliced together with frog genes until harvest season comes along, and well, the farmer doesn't have to harvest any of the potatoes because they just hop into the potato bag all by themselves. Again, this is hyperbole, hopefully, and this is an exaggeration, but it kind of makes the point that if it is convenient for you, our objections may go out the window. All right, so that is recombinant DNA. Let's move on to the second one, which is gene isolation. Gene isolation. This is when a certain portion of DNA, and again, we remember what DNA is, deoxyribonucleic acid, when a certain portion of DNA is isolated or identified in a narrow segment in order to be removed or replaced. Again, many of these techniques are first and foremost utilized in the medical field in an attempt to correct some defect or deficiency in DNA itself. So gene isolation is used to find or isolate a certain portion of DNA in order to remove it or replace it. And in so doing, some interesting things can happen. This is a technique that's being used right now in new gene therapies, these medical procedures that are even today able to correct people's vision by isolating defective segments of DNA and replacing them with healthy segments of DNA they've actually been able to give people their sight back again. And we'll be watching some videos after this lesson to see what exactly I'm talking about here. So here's a diagram of an isolation, identification and isolation technique where there is a series of genes which are identified, and here they're using bacteria with plant cells and replicating genes as a result, and then boom, kind of using what's called a gene gun in order to insert them into the gene they want to replace. And then the end result in this particular example is a transgenic plant that is regenerated from a single transformed gene. So this is done not necessarily in the health field, but in the agricultural field in order to give rise to plants that are perhaps resistant to disease or grow with certain characteristics. But in either case, genetic engineering is the way to go in today's day and age. So we've gone over the first two techniques, and we're going to stop it here for today. So we have time to watch some of the videos that I want to show you regarding these first two techniques. So until the next time we meet, I'll say bye bye.